Good morning, everyone. Thanks for joining. We will wait one minute more to make sure everybody is connecting. Thank you. So, I'm gonna start. Good morning, everyone, and thanks for joining our webinar session. I'm Manada Rocha, European Life Science Supplier Relationship Manager at Aventor, and I am very pleased to welcome you to today's webinar, The Future of Cell Culture, Transforming 3D Cell Culture into Reliable and Translatable Science, given by our colleagues from Molecular Devices, where they will share how labs can gain confidence in complex experimental outcomes using AI-powered solutions and automated cell culture protocols. For your information and to be totally transparent, unfortunately, one of the speakers, Oksana, was not available today, so you will see a recorded version of the webinar. But Felix Spera is with us today. Felix Spera is a hardware engineer and application manager at Molecular Devices, and he will be there to answer your questions. But before we start the presentation, I would like to give you some information about the progress of the webinar. No need to write down anything nor to take notes. This webinar is being recorded, so you will receive the replay link by email following this session. As said, there will be a Q&A session following the presentation, but not a live one. And what I mean by that is that you can't ask your questions live. You will have to submit your questions by writing them within the question box you see at the top menu at the right-hand corner of the screen. You can write them at any time, even during the presentation, and the questions will be read and answered during the Q&A session or later by email if we are running out of time. One last point. There will be a survey at the end of this session. May I ask you to fill it out? This is really important for us to get your feedback in order to improve the quality of our webinars. And finally, if you have additional comments, requirements, you can as well contact us at webinar@aventorscience.com or in the chat box. This said, I will hand over to our speakers and wish you a good session and talk to you later.
Hello, everyone. Thank you for your introduction. So I will be talking today about how automation will solve the challenges of complex 3D biology. So while I am manager of assay development group, so I will be talking about 3D biology and assay development. And then my presentation will be followed by Felix Spear, my colleague, who will talk about our new instrument, which enable automation of cell culture. So one of the main challenges of biology and biotechnology now is that about 90% of drug candidates fail during clinical trials. This high failure rate can be explained, at least in part, by insufficiency of current biological assays that used for drug screening. So organoids are believed to become a game changer in drug discovery because they are becoming bridging a gap between traditional cell assays and also tissues and organs. So what are the organoids? Organoids are 3D biological microtissues that contain several types of cells. They represent complexity, organization, and tissue structure, and they resemble at least some aspect of tissue functionality. Organoids will improve translatability compared to traditional 2D methods, and they will be representative for human biology and more predictive. They also suitable for complex disease modeling, and also they have lower cost and higher throughput than animal models. So what are important use of organoids? Obviously for drug discovery and also screening, for toxicity assessment, for disease modeling, when disease genotypes could be mimicked either by using patient-derived tissues or by introducing CRISPR-generated mutations. They could be used for personalized medicine using patient-derived organoids, which would allow predict drug effects. But there are multiple pain points by using these complex biological systems. They are labor intensive, complex, long-term processes. Those processes are technically challenging and require high expertise from scientists. And there are also issues with reproducibility and scale up of those assays. So there is a general organoid workflow. Typically, organoid workflow starts from 2D preculture. Cell origin may come from primary sources like intestinal, lung, or kidney, which rebuild tissue in a culture. Example could be intestinal organoids, which comes from Cleaver's lab. Another source of organoids would be iPSC-derived cells. So they culture and develop into multiple cell type organoids with organized structure. Example could be methods derived in a Knoblich lab for brain organoids. Typically, cells precultured or expanded in 2D for some time, depending on specific workflow. So development of organoids takes at least 7 days or more. It can take several weeks or even several months. The most typical method of organoid formation is by culturing cells in matrigel domes, when cells premixed with matrigel and droplets are placed into multiple plate format. They form a dome. So then they differentiate, they need to be passaged, they need to be processed, and this long process provides multiple technical hurdles, especially for automation and scale up. But they also provide opportunity for method development for technology developing companies. So methods and instrumentation are still evolving to facilitate research and also enable high throughput screening. So what are the main pain points by using 3D organoids for drug screening? So first of all, it's on a beginning part where you use cell source. So in addition to traditional cell line, it is desirable to use stem cell derived cells to differentiate them into particular tissues. They also could be coming from patient biopsies or other primary sources. Those 3D organoid models need to efficiently develop disease-relevant phenotypes. 3D culture and expansion, cell expansion is another challenge in this process. So scientists struggle with increased number of projects where different diseases, different subset of diseases need to be tested. This time-consuming complex process takes labor and time. And also there are multiple challenges with organoid quality, maturity, and uniformity. Setup of 3D assay and processing also present multiple challenges, which assay scale-up, accuracy, and reproducibility. And then complex analytical readouts and data processing require new methods to be developed to take full advantage from complexity of these 3D biological phenotypes. So here comes phenotypic screening, functional readout, multiplexity, and high content screening. Fortunately, technologies developed to overcome those barriers. Complexity of variability of biology can be addressed by using iPSC-derived organoids and CRISPR technology to introduce disease-related mutation. Complexity of cell culture can be addressed by using consumables and new microplates. Long-term culture and complexity of cell handling can be addressed with process automation. Then complexity of information which comes from these structures can be addressed by using high-content imaging and new functional readouts. Finally, on analytical sides, there are developing methods for complex statistical analysis, which include principal component analysis or machine learning. So there is typical screening workflow for 3D organoids actually looks pretty much similar for traditional 2D culture. It also starts from multi-well plates and where you can seed organoids, in this case, maybe by using matrigel or another matrix. So which they grow into organoids, they can be treated with compounds and then stained, and then you can use high content imaging, typically confocal imaging to see through organoids and through matrigel, and then you can derive multiple measurements, which would characterize those objects, their size, their complexity or morphology, and also can characterize cells inside of those organoids. So there are multiple organoid models which we and others dealt with. So for example, lung organoids, intestinal organoids, brain organoids, liver spheroids, and also some complex structures which have functional activity like neurospheroids or cardiac organoids, also organoids representing diseases like breast cancer or colorectal cancer. So to make this process more efficient, we develop a different automation method for those number of different workflows. So we developed Organoid Innovation Center, which includes a number of different instruments, like plate readers, like imaging instruments, liquid handlers, also automated robotic arms with scheduling software, which will enable us to develop automation of cell culture, which include also cell monitoring, media exchanges, compound addition, and endpoint assay. Also image analysis and data processing. Very importantly, scheduling multiple processes and different steps of the processes. So while automation of different methods of cell culture been used for drug screening for a while, so we were focusing on developing of new methods which is specific for 3D biology. For example, here you can see how automation allowed to place DOMs in a matrigel and does it with higher speed and precision than you can do it manually. Also develop multiple methods for liquid handling, for compound addition, for staining, which would address those technical challenges which comes with increased complexity of methods. So also in this process, we use imaging at different steps of the process and also analysis on the fly, which include machine learning algorithms. So software allows, for example, to find organoids and characterize their number and also density or size. I will present several examples of different models for organoids. For example, here is an image of lung organoids, and you can see this complex structure, so it has cavities inside. So the organoid culture could be imaged either by using 3D imaging, so by using multiple slides, or projection image. So it allows to not only quantitate number of cells inside the organoids, but also define differences in morphology or cell structure. So different markers can be used to mark different organelles or different cell phenotypes, for example, mitochondria, phalloidin, nuclear markers, and also specific markers indicating differentiation. So we developed methods by using AI-based analysis in transmitted light, and software could be trained to recognize those organoids and also define their number, density, or area. So this is example represent light dead staining of organoids. So organoids could be treated with different agents, for example, some components which mimic effect of toxic compounds like anti-cancer drugs or inflammation reagents. So then the most simple method of analysis, so you can run by using a projection image, and then you can use application models like live dead to quantify number and percentage of live dead cells or cells with different phenotype. More complex method could be used for multi-step analysis. For example, in this case, you can see software first finds organoids and then finds organelles inside of those organoids. And then you can characterize number of different cells, different organoids, and also content of percentage of different cells in organoids. So in this example, you can see organoids treated with ibrutinib. So you can use live dead staining to identify fact of toxicity. And then we run 3D analysis, which allow to identify number and percentage of dead cells. So for this example, software first provides analysis of images, which this stack of image, and then converts it in a 3D space. And more importantly, it can provide measurements which could be used to characterize and quantitate effect of different compounds. For example, here is liver spheroids treated with different compounds, which shows liver toxicity. And you can quantitate this effect, and actually we characterize number of different readouts which could be used for analysis, and some of those measurements actually follow this four-parametric curve fit, when some other do not, but still you can use successfully different measurements to get more complete information about phenotypic changes. So in this example, you can see cancer spheroids, and you can actually define not only fact of compound effect, but also you can define whether it's cytostatic or cytotoxic effect. So in this example, we use patient-derived cancer organoids for triple negative tumors, and those were treated with cancer drug romidepsin, and you can quantitate effect by using live dead staining. So by using high content imaging, you can actually see the effect of different anti-cancer drug and characterize the effect and effective concentrations. So we run actually a library of approved anti-cancer drug by using this cell line of patient-derived tumor. And we use high content imaging method to define different morphological and functional effect of compounds. And also we use automation for cell handling and compound addition. And as a result, we tested different concentration, and we were able to find a handful of novel compounds which were not considered for treatment of this specific tumor. Also, we will successfully use the cell painting assay for more complex analysis. So in this example, you can see intestinal organoids. So you can stain them also with different markers and use different magnification to characterize their phenotypes. So we can use this assay to test the toxic effect of different compounds, for example, anti-cancer drugs. So you can provide this imaging and image analysis, which would allow to define and quantitate different drug effects. You can use also higher magnification to identify specific cell phenotype, for example, presence and content of serotonin secreted cells. So high content imaging can be also used to characterization of more complex biological changes. For example, here angiogenesis, and it can detect different angiogenic sprouts and identify their volume and content. Also, like in this example, it can follow tracking of T cells penetration. And so this complex image analysis allow to characterize structural and morphological effects, but there are also methods which allow to characterize functional effects, which is specifically valuable in some tissues which represent functional activity. Like here is cardiac spheroids derived from stem cells. So you can see different drugs which modulate this functional activity. And you can measure traces by using imaging or by using different instrument, flipper, and characterize this potential cardiotoxic effect of different compounds. Even more interestingly, you can use actually neurospheroids and the same method to characterize functional activity and see effect of different neuroactive and neurotoxic compound, and also characterize disease phenotype. For example, here you can see disease of Alzheimer related mutation, which provides a phenotype which is different from a control and also can show effect of different drugs. So now I would like to introduce our new instrument, CellXpress AI Automated Cell Culture System. This new device will enable scientists to maintain 24/7 schedule for multiple experiments for growing and scaling up different organoids and cell systems. It will take control over feeding and pathogen schedule and also allow to characterize effect of what happening with your cultures. And most interestingly, it will enable to automate cell culture decision making by using machine learning tools, by taking advantage from imaging capability of this device. So this device includes liquid handler, which can help with feeding, seeding, and pathogen of cells. And also it has automated incubator capable of holding number of plates with sufficient capacity to run multiple experiments. So it also has imager, which provides different channels, different magnification, and allow to observe phenotypic changes and just monitor culture over the time. So most interestingly, it has a software which enable to combine these different tasks and different parts of device. So it controls liquid handling, it controls imaging, it controls the incubator, it controls moving plates from, let's say, incubator to imager or from incubator to a liquid handler, and so enables to design and execute multiple experiments and different multiple steps of experiment. So to build your protocol to build your experiment, you can start with predefined phases, which, for example, has a control different steps of the workflow, like cell harvesting or cell seeding, cell feeding, or kind of passaging, and also imaging. So for this device, so far we demonstrated and developed protocol for a number of most commonly used 3D cell culture workflows, which include spheroids, tumoroids, which include iPSC maintenance and passaging, and which include intestinal organoids in a matrigel. So I will describe briefly the workflow for those for those experiments. For example, spheroid tumoroid assay workflow starts from tumor-derived cells or cell line, which plated into U-bottom multi-well plates. And then those cells form spheroids and say cultured in an instrument and monitor, and then you can do their staining, you can do drug effects, monitor drug effects, you can also analyze those images and define your next steps. So this protocol is kind of suitable not only for cancer application, but also for any other organoids or spheroids which doesn't require matrigel. For example, cardiac organoids or neurospheroids, which usually grown in a U-shape low attachment plates. So here you can see example of spheroids grown by instrument. So this is HCT 116 cells. And so those were monitored, they were kind of treated with compounds, and compound effects was measured by using staining and fluorescent imaging. So the organoids were stained by using combination of first calcium AM and ethidium homodimer, and then images were taken. So you can see already kind of those dependent phenotypic effects, and they were kind of measured and also quantitated by instrument. So you can see image of intact organoid or organoid treated with doxorubicin. So, and then instrument provides data visualization, so you can select different measurements, and you can do plots in real time and how the experiment progresses. So to evaluate effects of anti-cancer compounds, so we treated these organoids with different concentration of anti-cancer drugs, and then took images, and we ran analysis which include the ratio of intensity of live versus dead stain, and we were able to observe effect of different compounds. This different workflow describes the passaging of iPSC cells. So this is done in 6-well plates, and you can see how automated process starts with media exchange. So you can see this media containers on the side and tilted plate to enable better media exchange. So in a process, then images are taken, so you can see stem cell colony, and image analysis enables quantitation of those colonies and area, and also cell differentiation. And also another step of protocol enables passaging, which includes addition of releaser reagent, mixing cells, and replate it into freshly prepared cells. So you can see in this example kind of monitoring of area of this iPSCs. You can see passaging and then growth and then passaging again and continuation. So interestingly, so image-guided decision-making can trigger next step of the protocol, including, for example, reaching certain density or reaching certain degree of differentiation. So you can select in a software the operator, for example, specific measurement, and then it will trigger a next step, like, for example, passaging, or it may just inform the user about time to trigger the next step like manually, just pushing continue button. So here is example which shows cell segmentation for stem cell phenotype or for differentiation phenotype, which done by using machine learning algorithms in Encarta software, which is also embedded into the instrument. So this is another workflow example, intestinal organoids. So you can see mix of matrigel placed in a deep well block, and then it's matrigel domes seeded, and then kind of lead solidified, then media added. It's all these steps done automatically. So the maintenance steps include media exchanges and also imaging. And then this image analysis on a fly again kind of follows these steps like growing and then passaging and growing again and so on. And so the images of organoids can be monitored at desired time point. For example, we did it every 24 hours, and then instrument and software builds the plots which you can observe. And the instrument can also do automated passaging, which kind of many of you know is a challenging process. So we developed protocols which allow to automate this step. So the uniformity actually and reproducibility is increased by using these automated protocols and automated handling. So this is example of matrigel domes with organoids in 24 well format, and you can actually do the analysis on the fly, which allow to monitor this culture by using different readout methods. So you can also do classification of these intestinal organoids for different phenotype and define drug effects by using this machine learning based analysis. So our next step of development is to use colorectal organoids and develop automated process which would allow drug screening. So with this note, I will transfer presentation to my colleague, Felix, who will tell you more about this new instrument.
Thank you very much for the introduction. And I'm going to talk a little bit more about the hardware of the system and also some applications. When we started the development of the CellXpress.ai, we needed to combine many different workflows into a single machine. And I listed the guiding stars of our system development. The system should and is able to handle both stem cells and organoids. It is able to handle hydrogels, including matrigel. We have a built-in monitoring system, a wide field imaging system that is able to monitor both 2D and 3D objects over orders of spatial magnitudes. For example, we can acquire images of single stem cells, and we can move up the scale to organoids, which are many millimeters in diameter. In order to analyze the data, we need to have a very robust imaging analysis pipeline, and the system is using both classical image analysis software and also machine learning-based image segmentation and analysis tools. As we already heard from Dr. Chirenko, the system is able to make decisions based on imaging readouts to trigger, for example, passaging or other interactions with the cells and the system. And it is a multi-user, multi-experiment environment. During validation, for example, we were running 10 different experiments in parallel, and we were using 5 different model systems, including stem cells, iPSC-derived organoids, and adult stem cell-derived organoids. Initially, the CellXpress.ai is supporting 3 different model systems, which are stem cells, iPSC-derived stem cells, spheroids or tumoroids, and 3D organoids, which are in a matrix, for example, matrixel, and also objects which are not embedded in the matrix. And to show you that this is a real system, I put in this presentation a picture, how it looks like. This is one of our prototypes. And now I provide you with a preview what is underneath the hood. On the top of the system, we do have laminar flow modules to keep the air inside the liquid handling deck sterile and clean. We do, as we have heard, an incubation, an automatic incubator, an automatic imager, and 2 ports to send out plates to the outside world. One is at the left side of the instrument, and there is another one at the back of the system. And this allows the integration of the CellXpress.ai into a larger automated setup. What you can see here is a closer look at the liquid handling deck. We do have a lot of storage positions for both plates and tips. And we have a working deck. And this working deck can cool or heat your plates. We do have tilting systems for a more efficient media transfer. And we do have a smart media module that can cool and heat the media. It can steer the media. It's very flexible because the media troughs can be replaced with different adapters that can hold conical tubes or plates. Then there is a solid and a liquid waste. On the left side again, I mentioned that already, we do have an external output to send plates to the outside world and to integrate the system into a larger environment. And the guiding star of this deck layout was to have a system that can autonomously feed and passage 40 plates over an entire weekend, meaning it needs to do 3 feeding events and a single passaging event. So this is a closer look at the smart media module, which is here where the orange arrow is pointing at. We do have positions for 8 of these little troughs. Instead of this trough, as I mentioned, one can also put some plates as well at this position. We do have also bulk media storage, which can hold 2 times 2 liters. And via tubing in the flow-through heater, the media is brought to the deck and is ending up in these little troughs that you can see here on the deck. This again is an even closer look at the smart media module. Again, you can see the little media troughs, which hold up to 370 milliliters of media. They can come either insulated or without insulation. As I mentioned, there is the possibility to steer the media if this is required, and we do have different adapters to efficiently cool or warm plates that can be placed in these positions as well. One nice feature is that based on the scheduling software, the system does know when it would need some media, and it can preheat the media prior to usage and cooling down afterwards. And this is key to ensure a long hands-off time to the user. The system comes with 2 different incubator capacities. One incubator can hold up to 44 plates, and it's shown here on the left-hand side, and another incubator that can hold up to 154 plates. A feature that we built in is an automatic decontamination feature, and we are using hydrogen peroxide vapor to automatically decontaminate and clean the incubator. The liquid handling deck is sterilized by using UV lamps. As I mentioned already, or as it was mentioned, the system also has a built-in wide field imaging system. The plates are automatically brought into the imaging system by built-in robotics. And for easier maintenance, the imaging system is sitting on a drawer and can be pulled out of the system to clean for cleaning reasons or to replace objectives. The system is able to illuminate the sample with fluorescence light. We have wavelengths between 365 and 645 nanometer excitation. We have 6 different positions for filter cubes. In addition to the fluorescence illumination, we also have transmitted light illumination for label-free imaging. The autofocus is really fast. It is possible to acquire an entire 96 well plate with a single field of view within 3.5 minutes. We support objectives between 2x and 40x magnification, and the objectives are oriented in the starlight configuration, as it can be seen here on the right-hand side, to allow the image acquisition of corner wells, even with larger magnifications. The system also has an environmental control. We can control temperature and CO2 inside the imaging chamber. And due to the large camera and our good optical design, the system is able to acquire an entire 96 well with a single image of our 2x objectives or 4 images using the 4x objective. Now we'll move more into the applications of the CellXpress.ai system. As Dr. Sharenko already mentioned, the system is able to handle major chill and to create or generate adult stem cell derived organoid domes. And how this looks like can be seen here in this image. And the built-in imaging system is able to follow the organoid growth over time. And every little dot here is an intestinal organoid. And the system is able to identify the optimal passaging time point based on imaging data, and how this can be working is shown in these images. What we do, we acquire images every day. And at some point, the system decides that this, in this case at 96 hours, would be the optimal passaging point. And this can be done by segmenting the images and pulling out some measurements, in this case, area and intensity, or intensity and skewness. And at a certain threshold, the decision is made to perform a passaging event. And it was mentioned that we are using machine learning tools for image analysis. And in this example, we do have a nice small dome with intestinal organoids. It is acquired using the built-in imaging system. And the first step is to segment the images. Now we can see that every object is labeled with a blue color, which is the segmentation mask. And afterwards, we can classify the images based on phenotypic features. And every color represents a different class. These examples only show that we can do the segmentation and classification not only in a small dome, but also in a larger dome, like the one on the left-hand side, or that we can place multiple domes and do the image classification also in these circumstances. What is shown on this slide is a validation of the phenotypic image classification. On the left-hand side, we do see different classes of our intestinal organoids that are present in our culture. And on the right-hand side, we compared the predicted label with a user annotated data set. So we had a user or a scientist going through all the images and manually annotate these images. And these data was then benchmarked with what the software would predict. And if we have a diagonal, as it can be seen in this example, we do have a very good match between what the user did and what the software thought should be end up in every respective class, indicating that we have a very good phenotypic classification by our system. This is something we have seen. It shows that we not only can segment intestinal organoids, but also different features of a stem cell colony, in this case differentiated cells and the core colony itself. Moving a little bit further into the stem cell workflow, what the system would do, it would seed cells, monitor and feed the cells every day, and at some point it would make the decision whether or not to passage. And when we do a passaging event, or when we do something else with our plates inside CellXpress.ai, we talk about different phases. And a passaging event, for example, could be a phase, or a feeding event could be a phase. And what the user can do, they can chain different phases behind each other, right? In this case, we have 4 different phases, block A, B, C, and D. And each phase could be something different, like liquid handling. It could be image acquisition, media exchange, or whatever the user would configure. And the transition between these phases are driven by decision making. And decision making is based on image acquisition, imaging readouts, and one can chain and link different measurements and readouts together to come up with a very complex rule set that then would move between the phases. What the system is also able to do is to track objects, wells, and plates over time. And this feature is called cell journey. In this case, we do have a plate with 5 wells. And what was happening, we have a passaging event. Each well was passaged into 3 different wells, giving rise to much, basically to upscale our experiment. And afterwards, these 3 wells, or these wells, were upscaled again to have even more samples. And then we only fed and imaged our samples. And because there may be many plates in the incubator, this allows the user to go forth and back in time and keep track about the samples in the experiment. But we not only can have this graph visualization, as we call it, we can also follow the individual images over time during multiple passages. In this example, we have our source plate, then we have our passage number 1, our passage number 2, and there would be another passage that didn't fit on the screen. So it is possible for the user to follow every well over time and to know what is going on in the system. We do also have a nice graphical scheduling software to keep track of what is going on inside the system. Each line would represent a different experiment, and we have a little microscope for scheduled microscopy events and a little drop for liquid handling in events. And the length of the bar would indicate the time that is required for each individual step. And this is a very convenient tool to get an understanding what is going on, and also to identify some possible scheduling defects, or to let the user know that it might be a good idea to have some media in the back pocket to add it to the liquid handling deck if they realize that something is upcoming, like a bigger passaging or feeding event. And this example is to show that we not only can acquire small objects like stem cell colonies or stem cells or intestinal organoids, but also massive multi-millimeter large cerebral organoids. I have to admit that they were cleared, but still they were few millimeters in size, and they were acquired using the 2x objective of the CellXpress.ai. With this, I like to stop to finish my presentation and ask for questions.
Thank you. So, Q&A session. So if not done yet, please submit your questions within the question mark, the small icon with the question mark, and we will go through it. So first question. Felix, are you there? Can you hear me?
Hello, I
Hello,
can hear
hello.
you.
Good. Is it possible to run multiple experiments simultaneously?
Yeah, thank you very much for those questions. A really good question, and this is absolutely possible. During validation, we did run up to 10 experiments simultaneously on the system, and we used 4 different model systems in parallel. Yeah, absolutely. This is something we can do.
Very good. Another question here. Do you use alternatives for merger gel?
Right. We did run a lot of experiments using Caltrex. There was like, I can't remember, some time ago, a real crisis to get Magidril. So we moved on to Caltrex, and this was also possible to use on our system. So we tested both.
Okay. How many different media types can you store on a deck?
Right. And this links very nicely to the first question about the different experiments that can be run simultaneously. In order to do that, we need to be able to store a lot of different types of media on deck. And we have 8 different positions of media that can be stored on deck, and 2 times 2 liter bulk media bottles. So we can accommodate 10 different media. And the nice thing is that the media can be cooled and warmed up. So when the system knows that a certain media is going to be used in the future or in the near future, it's being warmed up. And after it was used, it's cooled down again, if cooling and heating is required for longer storage for this media type. So 10 different medias.
Thank you. Another one here. Which software is used for recognizing or masking organoids in a well?
So
And
we're
how
using,
accurate is it?
pardon me, what was it?
And how accurate is it?
Accurate. So we're using for object segmentation. That's the question. We are using, there's different methods how an object can be segmented. Either, and everything, all the different tools are built into our CellXpress.ai software package. And either it can be segmented by using a deep learning network that the user needs to train, or the system is shipped with a couple of different networks, and the user can modify the networks to refine them for their needs. In this case, a deep learning network is used to segment the objects. In addition to that, one can use Molecular Devices CME, custom model editor, to use a classical histogram-based approach for segmentation, like thresholds or watershed. These things are also possible.
Yeah, the question is the question I read was not complete, and there are some other question part of it. Can you use it on a microscope pictures, or is it connected to an imager such as CellXpress?
Oh, whether the software can be used as a standalone software for images generated by a different system. No, not the CellXpress software package, but Molecular Devices is offering Incada and MetaExpress, and these software packages then can be used to segment and analyze image-based data. So Incada is offering deep learning, machine learning-based image segmentation and classification, and MXE is offering histogram-based imaging segmentation capabilities, and both work very nicely together in Kata and MXE.
Thanks. Is it possible to integrate a centrifuge for end-of passaging?
Good question. So the CellXpress is not just a standalone system. It can be integrated into a larger environment, and especially for the intestinal organoid or for the adult stem cell-derived organoid passaging workflow, a centrifuge is required. And yes, a centrifuge can be seamlessly added to the system so that a plate is sent out of the system by the back port, moved to the centrifuge, and then brought back into the system. So centrifuges, the flipper, and an IXM is natively being can be natively being added to the CellXpress. So yes.
And one last question. What parts of the software are responsible for the AI portion?
That is it's two parts. It's the AI-based image segmentation, which is deep learning network. Then we have the clustering software package, the phenoglyphs, what we called for phenotypic classification. And then the decision making itself, which would represent AI on the system. So centered around decision making and image analysis is what makes the system AI. What makes up the AI part of the system.
Oh, another one, last one, I think. How long does it take to feed a plate on the CellXpress AI?
Pardon me, the sound quality is not very good.
Sorry, how long does it take to feed a plate on the CellXpress AI?
How long does it take to feed a plate on the CellXpress AI? So yes. For stem cells, for example, we're feeding 2 plates simultaneously, and I believe it's like 5 minutes for 2 plates.
Okay. I'm not seeing any other questions. Just wait a few minutes. Okay. Felix, thank you very much for your comprehensive presentation and for answering the questions. As said, this webinar has been recorded, so you will get the link by email later on. You can also access the replay via Avanto's website, where it will be posted and remain available for 1 year, so you can go through it at your own convenience in case you missed something. So I hope you find this webinar interesting and helpful, that it has met your expectations, and please do not forget to answer the short survey at the end. This said, thank you very much for your time and your attention, and I wish you a very good day. Thank you, Felix, and thanks to the More Advice team.
Thank you. Bye.